In this video, we will discuss about uh, an example multi-step process of cancer and what are the reasons or the carci carcinogens that are responsible for initiation and proliferation of cancers. So let us start with an example of colon carcinomas. As we have understand or know from the previous video that colon carcinogens is usually occurred in aged people, and the number of people that are treated or uh, died because of colon carcinomas are increased with increase in age. And the reason that we have assumed in previous video is that uh, since since the cancer formation is a multi-step process, it requires times and multiple mutation for the cancer to get reach to a point where it is harmful to the human beings or cancer to get initiated. So this is the example of colon carcinomas. Initially, this is the uh, epithelial cell, and you can see that there is a mutation occur in one cell. Right now, this is not harmful. It is not a malignant tumor. It is adenoma. We can consider it as adenoma. It is tumor that is which is known as tumor that is not cancer. And this is the blood vessel, and this is the connective tissue. And with time, the proliferation of this mutated cell will occur. This is the lymphatic vessel, and these are the connective tissue. These black lines. And with time, the proliferation of this mutated cell occurs. And when this proliferation increased, then it might form a multi-layer, and which is known as small adenoma. And this adenoma means uh, that does not spread to another neighboring cells, that does not spread to another neighboring cell at the same time, or we can say that tumor that is not cancer. And this proliferation will increase with time, and that might lead to intermediate adenoma. Intermediate, at this point still it is not infectious, or it is been not in a position of metastasis. Metastasis is known as when a tumor is transferred from one part of the body to another part. Right now it is not in that state. However, when it further proliferation occur, and it forms a large adenoma plus carcinoma, or we can say that tumor, or we can say that initiation of metastasis. Here the mutated cell starts to move to other organelle. This is the blood vessel. They start to move to blood vessel. They starts to move to lymphatic vessel, and this invasion of blood and lymphatic vessel, and finally in this stage it is in the initiation of it is in the uh, initiation of metastasis. However, here the metastasis has uh, begin, and this is known as the carcinoma stage, or we can say that cancer state, where people get tumor in different part of the body, depending upon the path this mutated cell are spread across. So what is the reason for this mutation? The reason for these mutations are the carcinogens. The main root cause for cancer are carcinogens. So carcinogens are divided into two parts. First, that are the initiating agent that initiate mutation in the target gene, and result in result in the cell mutated cell that result in the mutated cell and which will proliferate with time and reach to a state where it is harmful or fatal to the host. So here are some of the carcinogens that might initiate the mutation. This benzoaphene, nickel carbonyl, and dimethyl introsamine. These are usually uh, intake with intake by the people who smokes frequently, because these are found in the smoke infected people. And this is aflatoxin. Aflatoxin is usually present in the molds that are that result in the mutation if it is not properly stored. That Let me repeat it. Aflatoxin is usually present in the molds that are used to store peanuts and other grains. Insufficient or inefficient storage of uh, inefficient storage of this uh, inefficient storage of these peanuts and malts may result in mutation of that malts that might create, or changes in the malts that might create aflatoxin, and that might inhale by the human beings along with that peanuts and grains. So these are just a few examples that might initiate mutation in the target gene or in the cells. However, this is just one type of carcinogens. We will see uh, the next type of carcinogens. So another type of carcinogens are those that promote cell proliferation. Uh, and this is one of the examples that are usually been carried out in the labs to artificially generate cancer uh, in the mouse or any other animal. And example of this carcinogen is phorbolesters that stimulates proliferation by activating protein kinase C signal. And this is the example we will see in detail, or see in some slightly more in depth about protein kinase C. But first we will understand this example. In this example, the mutation is occurred, and the cell is and mutated cell is formed inside the mouse. However, without this carcinogen that promotes cell proliferation, it is not feasible, or it will take a long time, or it is or even that cell might die, and people might not see any cancer initiation unless this kind of carcinogens are also being used in the experiment. So what this carcinogens does is it initiate or it promotes the proliferation of the mutated cell, and but they need to direct it in a way that it only proliferate mutated cell, uh, and this proliferation might result in tumor that might take a long time for generation if this carcinogen has not been used in the experiment. So this is though it is used here to accelerate the rate of cancer formation, it is also been used, or it is also been also take place during natural cancer formation process. It also take place a important role during natural cancer formation process in a host. So what is this protein kinase C? So whenever this signal is activated, what it will does is it will initiate the mutation. It will initiate the possibility of mutation. At the same time, it will prevent the cell from apoptosis. Apoptosis is the programmed cell death, and promotes its cell survival. So instead of killing those tumor cell, it will promote its cell survival, this protein kinase C pathway. And another thing that it does is it metastasis. It promotes metastasis. Metastasis is the transfer of tumor cells from one place to another place, or one part of the body to another part of the body. At the same time, it also promotes EMT migration, uh, anechosis resistance. EMT is uh, just the transformation of epithelial cell to mesenchymal cell. So because of this, the contraction of muscle occurs, and the chances of metastasis increases because the distance between the cells decreases. And the anechosis resistance is similar to apoptosis. It is anechosis is the program is known as program cell death, and um, the PKC promotes its resistance. That is, it's avoid the program cell death of these tumors, and and this signal makes the cancer therapy less effective, or cancer therapy is less resistance because of uh, cancer therapies are not much effective because of this protein C kinase, because it as it promotes the regrow, it promotes the cancer regeneration with the help of this like cancer mutation, or the metastasis, or the combination of both the process. So that is the reason this protein kinase C is considered as one of the important signaling pathway uh, in cancer study. So that's all for this video. Thank you for your time.